Thanks for that wonderful introduction and for the opportunity to present my work on engineering the core fusion machinery of SARS-CoV-2 spike protein as a proof of concept for vaccine immunogen design. So SARS-CoV-2 is an RNA virus that has spike proteins on its surface. So the receptor binding domain of spike attaches to the host cell receptor, ACE2, and spike undergoes dramatic conformational changes so that it transitions from the pre-fusion conformation to the post-fusion state. In the post-fusion conformation, the RBD is shed and the fusion peptide inserts into the host cell membrane. Then, the fusion machinery of spike brings together the virus and host cell membrane to fuse so that the virus can enter the cell and replicate. When we get vaccinated or infected with SARS-CoV-2, our adaptive immune system generates antibodies against spike as a protective mechanism. So antibodies can bind to different regions of the spike protein, such as the N-terminal domain, the receptor binding domain, and the more conserved stem domain. So antibodies that bind to the RBD are usually neutralizing because they prevent the virus from attaching to the host cell membrane, to the host cell receptor and subsequent viral entry and replication. Also, RBD antibodies are a correlate of protection from SARS-CoV-2 infection and severe outcomes. Therefore, a primary goal of vaccination is to elicit antibodies that target the RBD. So to make an effective vaccine, spike must be in its pre-fusion conformation, where the RBD is intact to generate RBD antibodies that will neutralize the virus. So previous studies on the first SARS coronavirus and MERS coronavirus spike proteins showed that mutating the hinge region of the core fusion machinery into proline mutations locks the spike protein into its pre-fusion conformation. These proline mutations rigidify the hinge and prevent the spike protein from transitioning to the extended post-fusion conformation. We were able to rapidly make effective vaccines against SARS-CoV-2 because the spike proteins of the first SARS coronavirus, MERS coronavirus, and SARS-CoV-2 are highly similar. However, identifying these two proline mutations was a laborious and time-intensive process that involved traditional protein design techniques. So to engineer immunogens for vaccine design, a structure of the immunogen must first be determined. Then, using the structure, one would identify mutations that they think would stabilize the pre-fusion conformation of the protein. So a few protein variants are recombinantly expressed and tested whether they are stabilized in their pre-fusion conformation through biochemical and biophysical assays. We want to improve the speed of vaccine design by targeting these steps um, of the process so that vaccines can be deployed earlier and faster. The current vaccine design pipeline is slow in throughput and biased because only a few mutations are selected and tested. It is also time intensive because identifying mutations, expressing, and purifying recombinant proteins usually takes at least 2 weeks or even longer depending on your lab's capacity. So to overcome the challenges of current vaccine immunogen design techniques, we developed a systematic high-throughput um, method based on deep mutational scanning to screen for pre-fusion stabilizing mutations. Our method allowed us to simultaneously quantify the cell surface expression and fusogenicity of thousands of spike protein mutations. We performed a deep mutational scan of the core fusion machinery because this is the region of the spike protein that is responsible for the switch from the pre-fusion conformation to the post-fusion conformation. We created a cell-based library of spike protein variants using landing pad cell technology. So each cell has one copy of the spike gene stably integrated into its genome and the corresponding spike mutant expressed on the cell surface, which establishes a genotype to phenotype link. These cells also express one half of a split green fluorescent protein intracellularly, which will be useful later on for our fusion assay. We measured the cell surface expression of spike protein through FACS sorting and performed deep sequencing to identify sequences that are high-expressing variants. Then, we created another stable cell line that expresses ACE2 on the cell surface and the other half of the green fluorescent protein intracellularly. We mixed the spike-expressing cell library with ACE2-expressing cells. If the spike protein is fusogenic and is expressed on the cell surface, then it will fuse with ACE2 expressing cells to form green fluorescing syncytium. On the other hand, if the spike mutant does not express on the cell surface, or if it is locked in this prefusion conformation, then it will not form green fluorescing syncytium. After co-culturing the cells, we collected two populations of cells based on green fluorescent signal and performed deep sequencing. We plotted the expression and fusogenicity of each mutant. A higher expression score means that the spike mutant is more abundantly expressed on the cell surface, and the higher fusion score means that the spike mutant is more fusogenic. For vaccine design purposes, we want a high expression score and a low fusion score. We first located that the K986P and the V987P mutations are in this region of the plot. These two mutations, which are found in current vaccines, serve as positive control and suggest that our screening method and strategy works. We identified additional prefusion stabilizing mutations that are indicated as non-fusogenic and proceeded to validate them using flow cytometry. So first, we verified that spike with these individual mutations express at higher levels on the cell surface compared to wild-type spike, and that these point mutations do not allow spike to fuse with ACE2 expressing cells. We then made various combinations of these prefusion stabilizing mutations and found that they express highly on the cell surface relative to wild-type and that they do not fuse with ACE2 expressing cells. We investigated how one of these mutations, aside from the two proline mutations, stabilized spike in its prefusion form through biochemical assays. So we proceeded to characterize the D994Q mutation. First, we expressed soluble versions of spike with the two proline mutations indicated by 2P and spike with the proline mutations and an additional D994Q mutation indicated as 2PQ. Thermal stability assays showed that both 2P and 2PQ have an apparent melting temperature of 47 degrees as indicated by the first peak. Interestingly, 2PQ has an additional peak at 62 degrees, suggesting that the additional D994Q mutation prevents the immediate complete unfolding of spike. We then determined the structural mechanism of this observation. So in an existing structure of spike with a 2P mutation, aspartate is unable to form an effective hydrogen bond with glutamine within the same protomer. When we performed modeling of the D994Q mutation, Q994 can form an effective hydrogen bond with glutamine within the same protomer. So this additional intraprotomer hydrogen bond can improve the thermal stability of spike protein by preventing its immediate complete unfolding. So from our study, we have established a systematic and high throughput method to identify mutations for effective vaccine immunogen design. However, to design effective universal vaccines, we need to determine whether mutations in the viral glycoprotein will weaken binding to protective antibodies. So mutations that weaken binding to antibodies pose a challenge in developing universal vaccines because this means that we need to take repeated boosters to keep up with a rapidly evolving pathogen. So in the case of SARS-CoV-2, current variants have several mutations in the N-terminal domain and receptor binding domain that prevent prior antibodies from neutralizing the virus. However, the spike protein also has a highly conserved stalk region that contains the core fusion machinery, which serves as an attractive target for broadly neutralizing antibodies that can potentially cross-react to newer variants. So we wanted to find out whether antibodies that target the core fusion machinery are effective in binding newer variants. So in a previous study, the authors showed that there is a common class of spike antibodies frequently found among vaccinees and patients. And this common class of antibodies is a target for vaccine design because it means that many individuals can be protected. And they showed that this class of antibodies binds to the first haptid repeat in the core fusion machinery of spike protein. So we wondered whether current and potential spike mutants weaken the binding of spike protein to these three HR1 antibodies. So first, we asked whether these antibodies are protective in vivo, even though they are non-neutralizing in vitro. We collaborated with Stanley Perlman and Roy Wong to show that these HR1 antibodies protect mice when challenged with mouse-adapted SARS coronavirus. We then mutated the Fc region of one of the antibodies and showed that the Fc region mediated the protection of mice from severe outcomes through antibody-dependent cellular cytotoxicity. We then asked which mutations in spike protein can weaken its binding to these protective antibodies. We performed FACS sorting using the spike mutant library that we created earlier and one of the HR1 antibodies as probes for binding. So we plotted the binding score and expression score for each mutation. A lower binding score means that the mutation decreases binding of spike to the antibody. D950N and Q954H, which are found in the Delta and Omicron variants respectively, weaken binding of spike to these antibodies. And we validated this finding using flow cytometry. This is one reason why antibodies generated from infection with earlier strains or vaccination with only the original spike protein does not fully protect the host from newer variants of the virus. To conclude, we have developed and established a high-throughput method to rapidly select for mutations that stabilize the pre-fusion conformation of spike protein in an unbiased and systematic manner. So this fusion and expression dataset that we have here is also useful for tracking potential mutations that might arise in nature. And we showed that mutations in the core fusion machinery, even though it is highly conserved, weaken the binding to public antibodies that are protective, which poses a challenge for universal coronavirus vaccine development. So this technology that we have described can be applied to developing vaccines for clinically significant infectious diseases that do not have vaccines yet, such as Lassa virus and respiratory syncytial virus. I would like to thank the people that have supported me in these projects that we have presented, in particular my advisor, Nicholas, for his support and for giving me the freedom to pursue my interests, as well as to Kenny for helping us set up the landing pad system that we have in the lab. And finally, we have an interactive data set where you're able to explore more into the fusion and binding and expression data that we have presented in this talk. Thank you, and I'm happy to answer any questions.
Thanks, Tim. Questions?
Thanks. Is your fusogenic assay, is that sensitive to proteases like TMPRSS2?
So for our cell line, the HEC293T landing pad does not endogenously express TMPRSS2. Yeah, so for our fusion assay, it is not sensitive to TMPRSS2.
How about for the ACE2 expressing cells? You're not exogenously expressing TMPRSS2?
Yes, so we had to insert the ACE2 gene in order to be exogenously expressed on the cell surface of the HEC293T landing pad cells that we have.
And then the one other thing I was wondering is, in your expression assays, I mean, I imagine this is something in which the promoter that you use could have a big effect. If the promoter is too strong, you could like clog up the ER. So how did you handle that? Have you tried different promoters?
So we did not try different promoters. So like what Kenny mentioned earlier, yes, definitely the promoter can have like the COSAC sequence can also have a huge effect on the cell surface expression of these proteins, but we did not test this in this assay.
Okay, thanks.
Thank you. Very interesting approach. Can you maybe say something about the timeline that all of this will take? So can you only apply this to viruses that are already around and have been studied for many years as the examples that you are mentioning? Or if the next pandemic hits us, should we all turn to you to find the perfect vaccine?
Yeah, so that's a great question. So one limitation and one really big limitation of our assay here is that we can only do this if we know the receptor of the virus. So if we do not know the receptor of the virus, then there is no way that we are able to do this fusion expression assay. Yeah, but as for the timeline that you mentioned, constructing the library and then performing the deep mutational scan, it can take as short as like a month, depending on your institutions and your lab's capacity.
Oh, great talk. Thank you. So I noticed that when you're trying to draw the correlation between your fusion score and your expression score, you just draw a line like crossing like 0 with like with slope 1. So how do you make sure that like your two experiments, like two facts, I don't know, expression experiments are on the same scale so you can make this assumption?
Yeah, so yeah, we performed independent replicates of these expression and fusion assays. And so our main assumption for like drawing that straight line, straight diagonal line in the fusion expression plot is that a particular variant that expresses highly would also be more fusogenic. And the converse is also true where a lowly expressing variant would also not be fusogenic.
So were you only looking at libraries of single variants, or is it worth thinking about combinations of variants if you want to have a bigger search space and things that have a bigger effect?
Yeah, so that's definitely a very valid question. So in our screen here, we only looked at single mutants because if we expanded it to double or triple mutations, the library size would be really big and intractable, and that would cause a lot of noise in when we process the data. So yeah, so what we do here is we just take the single mutants and then do the combinations afterwards.
Have you considered looking at any like ML models for variant effect prediction using these proteins?
So, oh, sorry, your question is on ML models.
Essentially, have you tried to see if some models trained on the spike protein have a high prediction for these variants? And then have you considered using these models to recommend potential other variants for your screen?
Yeah, that's a great question. So for my case here, we have not really considered machine learning models since this is primarily like a screen for fusogenic mutations. So we hope that this data set can actually inform and train machine learning models in the future.
All right then, well, if that's it, thank you, Tim, for a really interesting talk.